Gram staining procedure. The gram staining procedure was developed by Christian Gram in 1884. Differences in cell wall structure are one of the characteristic feature in identifying prokaryotes by a staining technique. This staining technique has been developed by Christian Gram in 1884. He is a Danish scientist. Based on the outcome of the staining procedure, prokaryotes may be categorized as gram positive or gram negative. Those bacteria have additional membrane outside the cell wall fall under the gram negative category. How we can identify bacteria from gram negative from gram positive? So this staining procedure is very important. Gram positive bacteria, those bacteria that retain the purple crystal violet dye when subjected to the gram staining procedure, because those bacteria have a thick cell wall approximately 15 to 80 nanometers consisting of several layers, as many as 40 layers of peptidoglycan, isn't it? But in the case of gram negative bacteria, and also in gram positive bacteria also have an additional molecule called teichoic acid, which run across the different proteoglycan layers, because this teichoic acid is combined with the peptidoglycan layer, which run across the different peptidoglycan layers. This may also be linked to membrane lipids, in which case these are known as lipoteichoic acids. This is the teichoic acid, and these teichoic acids may be linked with the membrane lipid. So in such cases, these are known as lipoteichoic acids. In the case of gram negative bacteria, the bacteria do not retain the crystal violet because these bacteria possess a relatively thin cell wall, approximately 10 nanometers. Gram positive bacteria have a thick cell wall, approximately 50 to 80 nanometers, consisting of several, that is 40 layers of peptidoglycan, and also these peptidoglycan layers and lipid layers are combined, form a cross-linked chain known as lipoteichoic acid. But in gram negative bacteria, relatively thin cell wall, approximately 10 nanometer, which is composed of a single layer of peptidoglycan surrounded by an additional membrane structure called outer membrane. The outer membrane of gram negative bacteria invariably contain a unique component, that is a lipopolysaccharide or LPS or endotoxin, which is highly toxic to animals. In gram negative bacteria, the outer membrane is usually thought as a part of cell wall. So gram negative gram stain will determine if antibiotic will work or if they will not. In a bacterial colony, we apply the gram staining procedure. The bacteria or the bacterial members are sorted out into gram positive bacteria and gram negative bacteria. After analyzing the characteristics of the bacteria and decided which antibiotic will work. Okay, so this is the staining procedure, Gram staining procedure developed by Christian Gram in 1884. So this is a very important technique. And how will be selected proper antibiotic? The proper antibiotic can be selected based on the Gram staining procedure. Again, Gram stain is the differential staining procedure producing different results because the bacterial colony or the bacteria can be identified based on the staining procedure. In the crystal violet, this is the procedure of Gram staining. First, the bacteria has to be stained with the crystal violet, and then using iodine. So all bacteria are purple. Then acid alcohol washed out. After washing, Gram positive bacteria showing purple color and Gram negative bacteria are colorless. Here we understood that the purple colored bacteria are Gram positive and the colorless are Gram negative. And using safranin, another stain, the Gram positive bacteria are again purple color, stay in purple color, but Gram negative bacteria retain red color. The color from safranin. Safranin is the red colored stain. So we accurately or we clearly indicated this staining procedure, indicated that bacteria are divided into two categories based on the outer covering. So in the Gram staining procedure, Gram positive bacteria retain purple color and Gram negative bacteria are colorless. And those bacteria are again stained with the safranin. Gram positive bacteria again stay their purple color, but the colorless Gram negative bacteria retain the red color from the safranin stain. So this is the Gram staining procedure. And here this picture once again describe the cell wall structure, detailed cell wall structure of the Gram negative cell envelope. This is much more complicated than Gram positive because it contains lipid, phospholipid, lipid A, periplasmic spaces. The periplasmic spaces are fluid filled area between cell wall and outer membrane and other various protein porous. So the Gram negative bacteria can produce LPS, that is lipopolysaccharide or endotoxin, which is toxic to animals. So this bacteria can cause bacterial infections. This is the detailed structure of bacterial cell wall. And that's next we move on the topic on bacterial cell membrane. Bacterial cell membrane, the importance of bacterial cell membrane. Bacterial cell membrane regulate what moves in and out of the cytoplasm because the bacterial cell membrane helping the bacteria which particles entering inside the cytoplasm or out.